In Chapter 16, we will look at how genes are regulated in bacteria. The first question I want to talk about is why genes actually need to be regulated at all. So we've talked in the past about the fact that highly complex multicellular organisms have all of these different types of tissues, and the reason why these tissues are different from each other is that different genes are turned on. So what about bacteria? They need to respond to their environment. And so bacteria growing on this plate, they are provided certain nutrients, certain type of energy, and they need to be able to use that energy. If they encounter a different type of nutrients, they need to switch and use that other kind of nutrients for their energy. And they need to be able to respond by turning on the correct genes, which can actually break down that food that is available. So in prokaryotes and eukaryotes, there are multiple different levels in which genes are regulated. And if we focus in on the central dogma, right, so we have DNA to RNA, RNA to protein. There are multiple places in this central dogma where regulation can occur. Usually we just think about the regulation of transcription, right? Whether a gene is transcribed or not, and that is one of the more important mechanisms of gene regulation. But there's also post-transcriptional regulation, regulation of RNAs. There's in eukaryotes, we have a level even before transcription, which is really regulation of how tightly that DNA is packed by the nucleosomes and whether it's accessible to be transcribed at all. We also have regulation in eukaryotes at the level of messenger RNA processing. And then at the level of RNA stability, whether it is accessible to be translated. And finally, we can have gene regulation at the protein level as well, where you might have a phosphorylation event that will make the protein active, whereas it's inactive without that phosphorylation. We're going to focus on regulation of transcription first, and I'd like to talk a little bit about what we call transcription factors. Transcription factors are proteins which will bind to regulatory sequences near the promoter, and there are a number of kinds of transcription factors that exist. There are multiple kinds of proteins. Helix turn helix is a very common type of protein motif that is able to bind directly to DNA. These two helices will actually bind in the major groove of DNA, and it's separated by that turn. Zinc finger is another kind of protein motif that is a DNA binding domain. This has these two zinc fingers, which again bind in adjacent areas of the major groove of the DNA. And then the last one is the leucine zipper. This kind of fits into the helix there as it's shown. So every transcription factor has a preferred sequence to which that protein will bind. That would be the consensus sequence, and this will not form covalent bonds, but it will form hydrogen bonds with the bases in the DNA. Transcription factors, they're also called transcriptional regulators or regulator proteins. You'll see a couple of different names for them. We're now going to focus on prokaryotes. Transcription factors in prokaryotes can be either activators or repressors. When an activator transcription factor binds to the DNA, it will help to enhance transcription. And so here is an example of this positive regulation by a transcriptional activator. Activator will bind right near the promoter, and it enhances transcription by helping to recruit the RNA polymerase to that promoter. In contrast, if you do not have the activator, sometimes that promoter is not enough all by itself to get the RNA polymerase recruited efficiently, and transcription won't occur. We can also have regulation by repressor proteins. So repressor proteins in prokaryotes specifically bind to a sequence called the operator sequence. When it binds to that sequence, it will actually inhibit the binding of RNA polymerase and the beginning of transcription. When the repressor is not there, you might be able to transcribe. Now, some genes will have both positive and negative regulation. Other genes may only have just one or the other, either an activator or a repressor that regulates it. Something else that I mentioned bacteria have to do is they have to be able to respond to an environmental stimulus. So we will talk about the lac operon as an example of a prokaryotic transcriptional regulation. The lac operon responds to a sugar, lactose, that it will find in its environment. So lactose or other types of sugars for other genes would be called effectors. Effectors are molecules from the environment that a cell can respond to. And so here is an example of what an effector might do. When an effector is present for an activator protein, it could be that the effector binds to that activator and changes the conformation of that activator protein to make it now able to bind to the activator binding site, and it will help turn on transcription. Another thing that might happen is that effector might bind to a repressor protein and it might change its conformation. In this case, it's showing an example where the repressor will bind to the operator in the absence of the effector, and when the effector is present, it changes the shape of that transcriptional repressor and it will make that protein fall off of the DNA, so it loses its affinity for the DNA. In both of these cases, the effector is actually an inducer, but I just want to point out that effectors can also be repressors for some genes. One other thing I need to introduce is an operon. In prokaryotes, a lot of genes are found that you have multiple genes that are regulated all by the same promoter and regulatory elements. So here's an example. This is an example in which all of these 5 genes are important for synthesizing the amino acid tryptophan. So they're functionally related genes, and all of them are regulated by the very same promoter. And so here would be the promoter, and there's an operator as well. And when that operon is transcribed, what happens is that that entire set of 5 genes is transcribed all in one messenger RNA. Those genes are then individually translated into different proteins. So now we're going to go into the lac operon. So we talk about the lac operon for a couple of reasons. First of all, it was actually the very first gene that it was discovered how its transcription is regulated. And second, it has both an activator and a repressor, and thus is very much a prototypical example of how other genes in bacteria are also regulated. I just want to point out that every single gene is regulated in its own distinct way. And so this is really just looking at an example to understand the basics of the components of transcriptional regulation. The lac operon has 3 different genes. So there's permease, beta-galactosidase, and transacetylase. These are the protein names. The genes that encode those proteins are the lacY, the lacZ, and the lacA genes. And these proteins are important for utilizing lactose, the sugar lactose, as an energy source. So permease is required to actively transport lactose into the cell. So we can get a little bit of lactose in there without that permease, but you need this protein to really get it into the cell efficiently. The second one is beta-galactosidase, and beta-galactosidase breaks down lactose, which is a 2-ring sugar, into 2 simple sugars. Then finally, the third gene, transacetylase, it's actually not clear whether or how it is involved in lactose metabolism, but it is regulated together with these other 2, so people assume that it has something to do with breaking down lactose. Now, this whole system was discovered by these 2 folks, Jakob and Monod, and they did some really elegant experiments. We're going to go into some of those experiments in class. They figured out how this operon works before DNA sequencing was invented. And so they really just used genetics. They induced mutations in the lac operon, and they observed how different mutations would affect the regulation of transcription of that operon. And then they proposed how it works based on this mutant approach. So this operon has 3 genes. So the lacZ, Y, and A genes are all made on this one RNA, which is then used to translate those 3 proteins. Right before the lacZ gene is a region of the DNA, which is the promoter, and we call that the lacP, so P for promoter. When RNA polymerase gets recruited to the promoter, it binds here and it will just transcribe all of those. The lacZ, Y, and A genes are also called structural genes. These are the genes that we really want to understand how they are regulated. Now, right next to that lac operon is the lacI gene. The lacI gene is a completely separate gene from these operon genes, and it actually has its own promoter. So it's regulated independently, and it is always transcribed. This lacI gene encodes a transcription factor that is a repressor. So here we would transcribe that into RNA and then produce that protein, which can go and bind to the region near the promoter, a region that is called the operator. That region, that LACO region, is actually right at the start of transcription, or the +1 site, and it physically blocks transcription from occurring. So whenever that repressor is bound here, you're not going to get transcription of the structural genes, the lacZ, Y, and A genes. Looking more closely at that promoter region, the LACO region is a DNA sequence that actually partially overlaps with the promoter itself and partially overlaps with the very beginning of that first gene on the operon, the lacZ gene. Now, how does this operon actually respond to the presence of lactose? So when lactose is not present, these genes are not needed, right? We don't need to transcribe because there's no lactose to break down or to import into the cell. So that repressor will bind and it just stops all transcription of those genes. When lactose is present, lactose will bind to the repressor and it will prevent that repressor from binding to the operator. Here is that repressor. The repressor gets bound by lactose. It's actually bound by a modified version of lactose. It's called allolactose, but that's just a minor point that I'm not going to ask you to learn about in detail. And this binding of lactose to the repressor changes the conformation of that repressor so that it no longer has affinity for the operator. And so the repressor falls off of the operator, and now transcription can occur. So here RNA polymerase can now bind now that the repressor is gone, and you will transcribe those genes and you will produce those proteins, beta-galactosidase, permease, and transacetylase. This in turn will help to break down lactose in the cell and allows that bacterium to use lactose as an energy source.